All right, sorry about that. So in crossing over, we're trading little pieces of DNA from mom and dad's chromosomes so that each sex cell that's created in you is a mixture of your mom and dad's chromosomes. Yes, da. Good baby. Okay, so when we're talking about how chromosome numbers change, you start with your normal diploid number, you double it in interphase, cut it in half in meiosis 1, and then cut it half again in meiosis 2. So the pattern is N, 2N, N, half N. So I repeated that again for the fruit fly and the bison. So if you know that a bison's normal cells have 60, then you double that in interphase, cut it in half in meiosis 1, cut it in half again in meiosis 2. If you know that a fruit fly has 16 chromosomes after interphase, that means it started with 8, doubled to get to 16, cut in half to get to 8, cut in half again to get to 4. Advantages and disadvantages of sexual reproduction: genetic diversity, you get the best of both parents and you're not a clone of a parent, which is always great. Negatives are that you have to find a partner to reproduce, the process is slower, and it's a lot more complex. Genetic disorders that we looked at were Down syndrome, Edwards syndrome, Patau syndrome, and Klinefelter syndrome. They're all trisomies of having an extra chromosome. We also have monosomies when you're missing chromosomes. All of them are pretty drastic for the most part. Klinefelter syndrome results in sterility and some secondary sex characteristics. Edwards syndrome is typically fatal in the infant before they're ever born. And then Patau syndrome, trisomy 13, results in a lot of organ defects. Stem cells we just covered today. Remember that stem cells are a blank slate. They can be programmed to become or fix any other cell type, and they can be used to study diseases. Learning targets on this unit: explaining the sequence of the cell cycle. That's that little circle diagram, the M, G1, S, G2 phases. I can explain what occurs during each phase. We just went over that. I can explain why mitosis produces identical cells, because there's no crossover, there's no mixing, it's just one cell splitting in two. Number 4, I can explain advantages and disadvantages of sexual, and then number 5, asexual reproduction. Remember, asexual is just doing mitosis. You're making one of you into two of you. It's an exact copy. It's good because it's fast and you don't need a partner, but it's bad because you don't have any diversity. Mitosis to cancer, we talked about avoiding checkpoints and uncontrolled division. Stem cells are blank slates. They help us produce specialized tissues. They can become any kind of cell. Meiosis produces non-identical cells because of crossing over. Examples and variation, so that's mixing up chromosomes between mom and dad. I can interpret a karyotype to identify potential chromosomal disorders. That's what you did in your online lab, wherever you spotted an extra copy of a chromosome or a missing copy of a chromosome. Good luck to you on the test. I'll see you tomorrow.